Introductory Neuroscience and Neuroinstrumentation, the Inverse Problem, and EEG Source Localization. Now, this is a slightly complex and theoretical lecture, so please bear with me. Okay. So here we will consider EEG source localization techniques and the forward and inverse problems associated with it. What it means is that we are recording from the scalp. Now, which part of the brain, which brain areas contribute to EEG? Now, can we do that just from the scalp electrodes? Can we figure that out, which areas cause different EEG and ERP components? That's what this lecture is about. So, during the last 2 decades, our computers have become more and more powerful. The computers' computational techniques used by earlier neurophysiologists were hamstrung because they were very weak compared to what we have with us now. The computational power of an Android smartphone is many orders of magnitude more than what the early neuroscientists, when I say early neuroscientists, I'm talking about the people from the 50s and 60s who started using computers, what they had. Okay. So the thing is, we are recording from the top. Okay, all these green dots are electrode positions, and using these, can we work backward and see which brain areas contribute or cause these electrical activity on the brain. So this is EEG source localization. Okay. So the forward problem. So in engineering, physics, or applied math, modeling involves predicting the effects or results for a set of known parameters. We know what's happening, we develop equations, and we have a mathematical theory, and then we can predict how the data looks like so. On the right, you have estimated parameters that gives rise to a mathematical model of physical theory, and then you have prediction of data. So this is the forward problem. As long as you know all this, and this is reasonably good, your theory, you would have a unique solution and you'll be able to predict data. Okay, so this is forward modeling, or the forward problem. So you have your something called your head model, which describes the scalp, the bone, the meninges, the cerebrospinal fluid, and the brain. And you have different sensors, and then the model also describes the conductivity and geometry of the brain. And you have equivalent dipoles which you can assume occur, and then you have a prediction, and then with your how it's supposed to look like, and then with your magnetic or electrical EEG sensors, you can record and see how it looks like. And if there's a discrepancy, you change your model until it fits what is recorded. Your prediction fits what's recorded. So this is the forward problem. Okay. The inverse problem, on the other hand, is we use EEG data to infer which brain areas caused it. Okay. So you have the measured data, it's backward. You have the measured data, your EEG, then you have your mathematical model of physical theory, and then you predict the parameters causing it. So unlike the forward problem, there is no unique solution for the inverse problem. In theory, an infinite number of parameters might explain the same measurement data. For example, you're recording between 2 areas, okay, 2 electrodes, okay, and you get 1. And it could be anything. It could be 5 minus 4, it could be 10 minus 9, it could be 8 minus 7, so on and so forth. There's an infinite possibilities. So that is why we don't have a unique solution. So what we do is we constrain the solution by using our knowledge of neuroanatomy and neurophysiology. For example, suppose we have the auditory evoked potential, and you have 2 possible solutions, one in the auditory cortex and one in the occipital cortex. So since we know the anatomy and the physiology, we would discard the occipital dipole or the occipital generator and focus on the auditory generator. Okay. So this is hardcore physics and numerical analysis with help from neuroanatomy and neurophysiology. Okay. And the more number of electrodes you have, the more robust your solution is. So suppose you just have 4 or 5 electrodes compared to 256 electrodes. So the 256 electrode data set would give you a much better generator solution than just a diminished set of electrodes. Okay. So assumptions. So there are a lot of assumptions which we use. Assumptions, physics, and conductivity and structure. When we are doing figuring out the sources and generators of EEG signals in the brain. So the first assumption is that the signal that is measured on the scalp is generated by the pyramidal neurons that are all oriented perpendicular to the cortical surface. And this figure you encountered earlier. So this is a pyramidal cell. One neuron generates a really a small bit of activity, and it cannot be picked up on the surface because all the other neurons around it they have activity and they overwhelm it, especially if they're not synchronized. But when a large group of neurons is simultaneously active, then it becomes an equivalent dipole. You can model it as that. It's a vector with direction and magnitude, and the electrical activity is big enough to be picked up by the EEG electrode on the scalp. So consider this figure here. You have the electrode over here. This is the scalp, the bone, the meninges, the cerebrospinal fluid, and then you have the cortex, and then you have the pyramidal cells in layers 2, 3, 5, and 6. So if they're all irregular, okay, and they're doing their own thing independently of each other, so you have these different electrical records from each, say, a neuron. The sum is the EEG and it's random. But if all of them are synchronized and they all fire, at the same time more or less, then the sum you can see, the sum potentials which reflect the summed activity of all these guys firing synchronously. As the apical dendrites of these pyramidal neurons are all parallel to each other, so the EPSP in these dendrites, they sum across space and time, and you have an approximate equivalent dipole which considers the activity of all these neurons. Okay? So the assumptions that the neural sources or dipoles exist only on the cortical surface and are perpendicular to it, it then imposes a constraint on the different kinds of solutions you would have for the inverse problem. So the solution search space, as it were, reduces to the cortical surface. You don't have to go inside or outside. And these constraints are then translated into a source model. It's very important to keep in mind that the final solution, what you get, depends on multiple parameters and the accuracy of the choices made. Because if the initial conditions or the initial parameters are slightly different, then the solution will be very different. So some of the things to keep in mind is we do a lot of simplifying assumptions. We assume that the current in dendrites flows unidirectionally, but actually that's not true, because if you remember, the action potential starts at the initial segment and it moves forward orthodromic, as well as it moves backward and invades the soma, the cell body. It's a back propagating action potentials. Then there's another whole population of cells which we haven't even talked about. Those are called glial cells, and they significantly outnumber the neurons and modulate neural activity. For example, they actively buffer extracellular potassium, and if you look at their resting membrane potential, it's near the equilibration potential of potassium. Okay, so they are not included in the source models usually. Then conductivity. So the different parts of the brain, the scalp, the coverings of the brain, the scalp, the skull, the meninges, the CSF, all of them have different conductivities. It's not homogeneous, neither is it isotropic. So we have to put in numbers for each of these conductivity values. And one big unknown is we do not know the conductivity value of the skull, the human skull, even now, because whatever skulls we have is usually post-mortem, and that's not the real situation, and we cannot assume that the post-mortem skull's conductivity is the same as a living person's skull. And we also don't know, we're not very clear about the conductivity of the tissue between the brain and the scalp surface. Most models assume the skull to be homogeneous with the same conductivity in all directions. This is still yet to be proved. So if you have an impedance meter and you have access to a neurosurgeon, please get the impedance value of the skull and the tissue between the brain and scalp surface in humans. Okay. So source localization in EEG. So this has been one of the primary goals, to solve this inverse problem. Okay. So what this means is identifying where in the brain a particular type of activity originates based on the surface EEG. And this is useful particularly for conditions like epilepsy, because epilepsy, the neuron, you have this kind of jerking movement, rhythmic movements very fast, and they imply hypersynchronization of pyramidal neurons, for example, and they're all firing, together in synchrony. So it will be very useful to localize which part of the brain the epilepsy is originating, because depending on that, for example, if it's in the temporal lobe and it's temporal lobe epilepsy, the treatment is specific. It can vary for different kinds of epilepsy. Okay. Now let's get to the nitty-gritty. How do you construct a realistic head model? So to identify the source of an EEG, you need three things. One is you need the source model, then you need the head model, and then you actually need the EEG data. And there are different compartments which have to be individually modeled. One is the skin, then you have the bone, compact bone, as opposed to spongy bone. The compact bone is on the outer and inner surface of the skull. The spongy bone is in between. Then, of course, the gray matter with all its neurons and glial cells and what have you, and the white matter, which is made up of mostly axons, and the cerebrospinal fluid. Okay. So all these have to be put in, to do your source modeling. So the source model, let's consider the source model first. The source model tells us the three-dimensional positions, that is the x, y, z coordinates of the dipoles on the cortical surface. And this would be over here, over the gray. And this is also referred to as the source space. And it's assumed that the EEG signals are generated by sources which can be approximated as dipoles for reasons mentioned earlier. So then we come to the head model. We need to describe how the electrical currents from these sources will flow through the head and finally end up as scalp EEG. So this depends on two factors. One is the geometry of the head, and the other factor is the conductivity of various tissues of the head. So this is, skin, compact bone, spongy bone, all these would be involved for the head model. Okay. And the head model can be, the geometry of the head can be obtained by MRI, structural magnetic resonance imaging, not fMRI, just plain old anatomical MRI. Regarding the rest of the tissues, there's a huge literature available on conductivity values for the various tissues. But as mentioned before, skull conductivity is the biggest unknown. We assume. We don't have any clear experimental data for skull conductivity. Okay. So thank you. Okay.